Hello everyone, welcome back to my channel Microbiology. Today I am going to present an important human intestinal protozoan parasite, Entamoeba histolytica. Our topic is Entamoeba histolytica, its morphology, pathogenesis, and laboratory diagnosis. Now let's just start it. This parasite is the causative agent of amoebiasis, an important public health problem in many of the developing countries including Nepal. And the infection mainly occur due to the poor sanitation, contaminated food, and unsafe drinking water. During this presentation, we will discuss the introduction and its history, habitat and classification, morphological forms, its life cycle, pathogenesis and clinical features, laboratory diagnosis, treatment, and prevention. If you find the video useful, please don't forget to like, share, and subscribe my YouTube channel. Now let's discuss about the general introduction and its discovery. Entamoeba histolytica was discovered by Loss in 1875. He demonstrated the parasite in dysenteric fishes of a patient in Russia. Similarly, in 1890, William Osler reported the case of a young man with a dysentery who later on died of the liver abscesses. In this figure, we can see the several morphological feature of the Entamoeba histolytica showing the trophozoite cyst and nucleus. Now let's discuss in detail about Entamoeba histolytica. Okay. Entamoeba histolytica is a pathogenic protozoan parasite that primarily infect the large intestine of human. It's in a severe cases, it may spread to the extraintestinal organs such as the liver, lung, and brain. And amoebiasis may occur in the different form as a asymptomatic infection, intestinal amoebiasis or the amoebic dysentery, and extraintestinal amoebiasis such as the amoebic liver abscesses, and human are the only natural host of this parasite. And amoebiasis is the major public health problem in developing countries including Nepal due to the poor sanitation and contaminated food and water. Now moving to the habitat, Entamoeba histolytica lives in the mucosa and submucosa of the large intestine of human, and human are the only natural host. And the classification, talking about the classification, Entamoeba histolytica belongs to the kingdom Protista. Similarly, phylum Sarcomastigophora, subphylum Sarcodina, class Lobosia, order Amoebida, family Entamoebidae, genus Entamoeba, and the species Entamoeba histolytica. Morphologically, Entamoeba histolytica exists in a three form, that is trophozoite form, precystic form, and cyst form. In the figure, we can see the trophozoite figure of the Entamoeba histolytica in A. B indicate the precystic stage of the parasite, and C indicate the uninucleated cyst, with a D indicate the binucleated cyst, and the mature cyst of the Entamoeba histolytica is quadrinucleated as shown in the figure. Okay, now let's discuss each morphological form of the parasite in a detail. First is the first morphological form is trophozoite, which is also called as a vegetative form of the parasite. It is actively motile and only form present on the tissue and found in the fresh dysenteric stool in active cases and survive only for a few hours, it is also responsible for the tissue invasion as it is the only form present on the tissue. And the size vary from 10 to 60 micrometer and in average 20 to 30 micrometer in diameter with a irregular shape, and the cytoplasma of the trophozoite can be divided into a clear outer ectoplasma and inner granular endoplasma. The outer ectoplasma is clear hyaline outer layer, and the inner endoplasma is granular inner layer with a red blood cell leukocytes, and tissue debris together with the food vacuoles. The red blood cell may, however, absent if the infection is confined to the gut lumen. The trophozoites of the parasite is actively motile, and the motility is due to the presence of long finger-like projections of the ectoplasm into which endoplasm flows and known as a pseudopodia. And the inner nucleus is the parasite has a inner single nucleus which is spherical in a shape varying from 4 to 6 micrometer in a size, containing a central karyosome surrounded by a clear halo and anchored to the nuclear membrane by the fine radiating fibrils known as a Leydig network, giving it a cartwheel appearance. The nucleus is not clearly seen in the living trophozoites but can be clearly demonstrated in the preparation stained with the iron hematoxylin. And in the inner endoplasm, there is presence of a food vacuole which contain the ingested RBC. That it is also a diagnostic feature of the trophozoites, and trophozoite can survive up to the 5 hours, average in a 5 hour at the 37 degrees Celsius, and are easily killed by the drying heat and chemical sterilization. Therefore, the infection is not transmitted by the trophozoites. Even if the live trophozoites from the freshly passed stools are ingested, they are rapidly destroyed in our stomach and cannot initiate the infection because of the gastric pH. Next morphological feature is precyst. It is smaller than trophozoite, measuring 10 to 20 micrometer, rounded or oval in a shape, and it prepares for the incrustations. And it is non-motile without ingested RBC. The trophozoites undergo the encystment in the intestinal lumen. This encystment does not occur in the tissue or in a feces outside the body. So before encystment, the trophozoite extrudes its food vacuoles and become round or oval, about 10 to 20 micrometer in a size, and this is known as the precystic stage of the parasite. It contains a large glycogen vacuole and two chromatid bars. It then secretes a highly retractile cyst wall around it and becomes cyst. Now, cyst is the resistance. Cystic structure of the parasite is resistance to environment, and it is also the infective stage of the parasite found in the formed stool. The cyst is spherical in a shape, measuring about 10 to 20 micrometer in a size. It is non-motile, and the early cyst contain a single nucleus with a chromatid bodies with 1 to 4 chromatid bodies or a chromatidial bars or mass of the glycogen. And these chromatid bodies are cigar-shaped refractile rod with a rounded end, and as the cyst matures, the glycogen mass and the chromatidial bars disappears, and the nucleus of the cyst undergo the two successive mitotic division to form two and then four nuclei. And the mature cyst is thus quadrinucleated, and the cyst wall is highly refractile membrane. It makes it highly resistant to the gastric juice and uh, and and enable them to survive at an unfavorable environmental condition. Okay, now we will learn life cycle of the parasite in detail. The Entamoeba histolytica passes its life cycle only in a one human host, and the infective form is the mature quadrinucleated cyst passed in fishes of the convalescents and carriers. The cyst can remain viable under the moist conditions for about 10 days and act as an infective form of the parasite. And the human acquire the infection by fecal-oral route by consumption of the contaminated food, water, and raw vegetables. As the cyst wall is resistant to the action of the gastric juice, the cyst passes through the stomach and stomach undamaged and enter into the small intestine, and the process of excision occur. When the cyst reaches the cecum or the lower part of the ileum, due to the alkaline medium, the cyst wall is damaged by the trypsin leading to the excision, forming a metacyst. Here, the cytoplasm gets detached from the cyst wall and the amoeboid movement appear causing a tear in the cyst wall through which the quadrinucleated amoeba is liberated, and this stage is called as the metacystic stage of the parasites. And after the nuclei in the metacyst immediately undergo the division to form the eight nuclei, each of which gets surrounded by its own cytoplasm to form the eight small amoebule or metacystic trophozoites. And then, if the infestation takes place in the small intestine, the metacystic trophozoites does not colonize there but are carried to the cecum. And the optimal habitat for the metacystic trophozoite is the submucosal tissue of the cecum and the colon, where they lodge in the glandular crypts and grow by the binary fission. However, some develop into the precystic forms and cysts which are passed in the fishes to repeat the life cycle. The entire life cycle is thus completed in a single host. In most of the cases, the Entamoeba histolytica remained as a commensal in the large intestine without causing any ill effect, and such persons become a carrier or symptomatic cyst passers and are responsible for the maintenance of the spread of the infection in the community. Sometimes the infection may be activated and the clinical disease inserts such as latency and reactivations are the characteristics of the amoebiasis. Okay, now let's discuss the pathogenesis of the parasite. As I already mentioned, the mode of infection of the parasite is due to the fecal-oral route by consumption of the contaminated food, water, and vegetables. And Entamoeba histolytica causes the intestinal and extraintestinal amoebiasis, and incubation period is highly variable on average in range from the 4 days to the 4 months, and amoebiasis can present in the different form as a degree of the severity depending on the organ affected and the extent of the damage caused. And the intestinal amoebiasis, the pathogenesis of intestinal amoebiasis includes the initially the lumen-dwelling amoeba do not cause any illness as they cause the disease only when they invade the intestinal tissues. And this happen only in about 10% of cases of the infections, and the remaining 90% being the asymptomatic. And the parasite attached to the chronic mucosa by the amoebic lectins, and the trophozoites penetrate the intestinal epithelium using its motility, and the proteinases, cysteine proteinases, histolysine, and collagenase enzymes. And these enzymes cause the destruction of extracellular matrix, immunoglobulin A, and the complement protein, resulting the inflammatory reactions also contribute to the tissue damage. And the ulcer formation is the typical ulcer. It is the typical lesions in the flask shape ulcer is formed, and these ulcers are multiple or mainly found in the cecum and a sigmoid colon surrounded by the healthy mucosa. Clinical manifestations of intestinal amoebiasis may vary from the mild disease to the severe dysentery, and the common symptoms include the abdominal pain, diarrhea with a blood in a mucus, gradual onset of or usually no fever, and typical stool findings involve the foul-smelling stools with a blood and a mucus, trophozoites containing the RBC, and sometimes the charcoal-laden crystals can be found. And at a severe disease, it may lead to the fulminant colitis with a toxicity and other chronic relapsing infection. And sometimes the chronic sickle involvement may mimic the appendicitis. About 5% individual with the intestinal amoebiasis after 1 to 3 months after the disappearance of the dysentery attack may develop the hepatic amoebiasis and most commonly affect the liver. But it may also involve the lung, brain, skin, pericardium, and the most frequent form of the extraintestinal amoebiasis is the amoebic liver abscesses, which shows the clinical symptoms like fever, right upper abdominal pain, pain radiating to the right shoulder, weakness, weight loss, loss of appetite, and enlarged tender liver and intercostal tenderness. And the involvement of the lung or the pulmonary amoebiasis may produce the chest pain, cough, blood-stained sputum, and the cerebral amoebiasis is rare but is very dangerous and may cause the headache, vomiting, seizures, and paralysis in individual. And the complications may include the rupture of the liver abscesses, peritonitis, and pleural effusions and brain abscesses. Okay, now let's discuss about the laboratory diagnosis. The laboratory diagnosis of the intestinal amoebiasis. For the diagnosis of the intestinal amoebiasis, the most important diagnostic method is stool examination. The fresh stool should be examined immediately, and the macroscopic observation of the stool should be carried out for the semi-liquid brownish stool with a mixed with a blood and mucus, and the slime weight mount shows the motile trophozoites with ingested RBC and sometimes charcoal laden crystals, and the iodine preparation can be done to demonstrate the nucleus, glycogen mass, and the chromatoid bodies. And sometimes the permanent stains, such as a trichome stain or a iron hematoxylin stain, can be done to examine because the cyst excretion and at least three stool sample should be examined because the cyst excretion is intermittent, and the culture can also be done using the Balmont medium or a Cargill's medium, and the confirmatory finding is trophozoite containing the ingested RBC. And for the diagnosis of the extraintestinal amoebiasis, in a hepatic amoebiasis, the trophozoites are usually found at the edge of the liver abscesses and cysts are absent. So the several serological tests such as ELISA, indirect hemagglutination, latex agglutination, and countercurrent immunoelectrophoresis can be done, and several radiological investigations such as ultrasound, CT scan, and X-ray can also be done. And the ultrasound is commonly used to detect the liver abscesses. And after diagnosis, the treatment includes the omibicidial drugs and fluid and electrolyte replacement and blood transfusion if the severe blood loss has occurred, and a large liver abscesses may require the aspiration to prevent the rupture. And the prevention of the disease can mainly depends upon the good sanitation and hygiene, which is important, and the several important preventive measures such as safe disposal of the fishes, avoiding the contamination of the food and water, similarly, drinking the boiled or properly treated water, avoiding the use of a human feces as a fertilizer, treating the asymptomatic carriers, and proper health educations and food handlers with the infection should be treated to prevent the spread. Okay, now let's just quickly summarize about the parasite. The Entamoeba histolytica is an important pathogenic protozoan responsible for amoebiasis. It exists in trophozoite, persist, and cyst form, and cyst is the infective stage while the trophozoite cause the tissue invasion and disease. Early laboratory diagnosis and proper treatment are essential for the prevent and complication such as amoebic liver abscesses, and improved sanitation, safe drinking water, and hygiene remain the key method for the prevention of the disease. If you find this video useful, please don't forget to like, comment, share, and subscribe my YouTube channel. We in our next video we will discuss the similar educational aspect of some parasites. Till then, thank you for watching